Assalamu alaikum and welcome to the Dr. UUT channel. In this video, we will talk about the cell cycle, one of the cell cycles, mitosis. This one is in English and available in Urdu. You can search the lecture in Urdu also. Cell cycle and mitosis. What is mitosis? It is one of the cell cycles. First, we'll know the meaning of mitosis. Mitosis means thread formation. Now, why it is given the name as thread formation? In the ancient time, when the scientists were studying the cell, so they observed a kind of more thread formations. So the scientists in that time, they were calling the chromosomes the threads. Then it was changed to the mitosis. So mitosis means thread formation, in which what happened? According to us, we say chromosome. The chromosome duplication happened at that time. They were calling this chromosome as a thread. So this threading mechanism was observed. That's why it got the name. Well, coming to the point. Mitosis occurs in the body cell, and body cells are also called as somatic cells. The time that one cell takes to divide into two is about 10 to 24 hours. Means that one cell will divide into two cells. This will take the time about 10 to 24 hours. Means within 24 hours, a cell will divide into two. So a human body is covered with number of cells. Now every cell is dividing. The question is that yes, every cell is dividing in the body. Then why not the human body size increase? The reason is that when the cell divide, so the new cells will come up and the old cells which are present at the top, they will shed. Due to which what will happen? Every 24 hours there is shedding from the body of the cells, the old cells, and the new cells will come and will take the place of the old cells, due to which our size will not increase that much, but with the time our muscles grow and our bones grow, we will grow and our size would increase, but not due to this that much extensive cell division, and they are helping us to cover the body. Well, we can say in short that whenever after every 24 hours the new cells are formed and the old cells are just shed, and the time that I mentioned here, it is about 10 to 24 hours. Why not specific time? The reason is that in every eukaryotic cell, the cell divisions take place, but it depends upon the activity of that cell. The more the cell is active, the more will be the division time less. Example, if a person, I am standing in front of you, if I'm just speaking, the way I am doing action is different, and if someone is there who is doing exercise, jogging and anything else, so the time that the person will take the cell cycle, if I talk to you about the cell cycle time, my cell cycle will take about will be in between 10 to 24 hours, but the one who is doing exercise, jogging, everything, for him the cell cycle time will decrease. Because of what? Because he is enough active, or he's doing exercise more than me, and his cells are shed more quick than me. So for that, the new cell is supposed to be formed, and for him what will happen? This time will decrease. Means he might take about 10 to 15 or 20 hours, but I will take from 10 to 24. Means I will take a lot of time, because my cell cycle will be a bit in a slow mode. So that's it, due to which I mentioned that it will take 10 to 24 hours for one cell to divide into two. And now when a cell wants to divide into two, it has to double everything present inside the cell. That is actually called mitosis, in which what has happened? Doubling is there. Mitosis is called for the same reason, because threading were observed. You can say a normal human body contains, human cell, 46 chromosomes. When we divide 46 into half, it will come 23. So a human body is formed from two haploid number of chromosomes, 23 from the mother's side, 23 from the father's side. You can say sperm and egg. When they fuse, they will form the 46 chromosome. Now we are not talking about the meiosis, we are talking about the mitosis. We are not talking about the meiosis, but we are talking about the mitosis. In the mitosis, what will happen? That this 46, which is diploid, this diploid cell will undergo the process of cell cycle or division. After the division, what will happen? This cell will divide into two, and each cell will contain 46. Means each cell is now diploid. Question rises, how this one diploid is giving two diploid? Now when we plus both of them together, 46, you multiply them by 2. What will happen? you will get the number of chromosomes equal to 92. So now here just 46, and how 92 chromosomes are formed? The answer is simple. Mitosis means in which what was happening? Threading was happening, in which the chromosomes were duplicated. In a sense, you can say a replication of the DNA, due to which what was going to happen? One chromosome which was formed was actually doubled, because DNA is doubled. Well, we will understand in our further steps. First, we have to know about something, that when the DNA replicates, what happens? Where before it was supposed to form one chromosome, but now two chromosomes will be formed, and these chromosomes will be held by the centromere, the center protein. It will hold the sister chromatids. Now this is called sister chromatid, and this is also called sister chromatid. These chromatids are then you can say anchored or holded by a centromere. And beside the centromere, one thing you must know more is that there is another protein available known as kinetochore. Now, what is kinetochore? This is providing a kind of a holding place for the spindle fibers of the centrioles. The centrioles will just hold this. Now, what is this actually I'm talking about? We'll understand these things in our phases of the mitosis. So, coming back, when a cell wants to divide into two, it has to go in certain phases. The very first phase that it enters is known as interphase, which is also known as preparatory phase, or you can say it as preparation phase. In this, the cell will prepare itself for the division. So preparation phase is further composed of G1, S and G2 phase. G1 stands for gave 1, S for the synthesis phase, G2 for the gave 2 phase, and then it will enter into the mitosis. In the mitosis, we have certain more phases. After the mitosis, there will be cytokinesis, in which a cell will divide into two. Now, coming to explanation, G1. In the G1 phase of the interphase, everything present in the cell will divide. Everything. Just one thing will take the duplication, will undergo the duplication. That is the DNA, which will take place in the S phase. S phase, synthesis phase. In this phase, the DNA will replicate. Means copies are formed. After that, what will happen? Then the cell will enter into the G2 phase. In the G2 phase, what will happen is that all the enzymes present necessary for the cell division will be formed in the G2 phase. Everything is formed in the G1. DNA replication or synthesis takes place in the S phase or in the synthesis phase. In G2, all the enzymes are formed. Then the cell will enter into the M phase or mitosis. In the mitosis, we have prophase, metaphase, anaphase, telophase, and cytokinesis. After is the M phase. So in the prophase, what will happen? The DNA, when it is in uncoiled form, we call it as chromatin. The chromatin, and we have some certain types of proteins known as histone proteins. The DNA will combine with the histone protein, or it will coil on histone protein, so the chromosomes will be formed. You can say in the prophase, the chromosomes are formed. The centrioles present, they will start moving to the sides, and the nuclear membrane that was around the nucleus here, it will start disappearing, and Kinetochore may bring themselves available for the centrioles to attach. Centrioles will start attaching on the kinetochore, and like this, the centrioles will attach to a chromosome and will bring the chromosome at the center. In the prophase, chromosomes appeared, centrioles moved to the corners, spindle fibers appeared, nuclear envelope disappeared, kinetochores became available, and in the metaphase, then what happened? The centrioles brought these chromosomes at the equatorial line of the cell, known as metaphase plate. Metaphase plate. And in the meanwhile, a kind of force is exerted. Now, is being felt by the chromosomes. One centriole is pulling upward, another centriole is pulling it downward. So the chromosomes are now in the equatorial region. And what will happen? This protein that is holding these sister chromatids, known as centromere, what will happen to them? Degradation of the proteins holding the sister chromatids. Proteins that are holding the sister chromatids, that will degrade, means that will be broken. After that, the cell is in the tense condition. The centrioles are pulling it down and up. Centrioles are pulling it down, means each centriole is pulling the chromosome to its side. Like this, what will happen? The chromosomes will separate after the degradation in the anaphase. In the telophase, they will move. And like this, what will happen? In the middle of the cell, a ring is there of the actin filament. Actin filament. You know, actin myosin responsible for the contraction of the muscles. That actin will then cause the contraction. A kind of furrow means it will cleave. It will start cleaving, breaking the cell into two, and this breaking will complete in the cytokinesis. So what will happen? Then two cells will be formed, each containing the diploid number of chromosomes. Why? Because we got double duplication in synthesis process, means replication is done in the S phase, and now here in the equatorial region in the metaphase we were having 92 chromosomes aligned. So due to the pull, what happened in the anaphase, 46 moved up, 46 moved down. So then what happened in the telophase? Now 46 is present in the one cell, in the next cell after the cytokinesis. So both contains 46 and 46. Like this, diploid cells are formed in a sense, because I'm giving the example of the human body. So this is how the mitosis takes place in the living body, in the human organism. This happens in all, but here I mentioned about 46. If you remove the term 46 from this complete lecture, you can fit this lecture for every cell, then every eukaryotic cell. But if you are just bringing the lecture to the human body, then you are supposed to give the 46 number, or you are supposed to put the 46 in my lecture.